I'm Uta Francke. I'm a human geneticist, originally from Germany, as you can hear. And I'm an emeritus professor here at Stanford and also senior medical director at 23andMe.
So we have 3 billion base pairs in just one copy of the human genome, and we've learned that we have 2 copies of the human genome, of the nuclear genome in our cells, and we've been learning a bit about the coding regions, the protein coding regions. What can you tell us about the relative size of the protein coding regions of the genome versus the sort of non-coding regions?
The big question was, how many protein coding genes are there in the genome? And initially, before the Genome project, the number that's been kicked around was like 100,000, sort of a wild guess. And then between 2000 and 2003, there was an official betting game going on, where people could put down $1 and a number. And then years later, it was $5, and then it was $20, because then it became more difficult. And everybody could only put down one bet. About 150 people put down a bet, and the ranges of their guesses went from, you wouldn't believe it, 25,000 to 150,000.
Wow.
So nobody had really any good idea. The mean was around 61,000 genes. And when the first draft was published, they said, well, we had a brief look at it, and we think it's between 30 and 35,000. And when it was finally finished, the number had come down to 20 to 25,000. And many people were surprised, because this is hardly any more than the roundworm or the fruit fly. So the humans are so much more complex. Shouldn't they have more protein coding genes? So the complexity cannot be immediately deduced from the number of genes when you don't know what these genes can be used for.
So what percentage of the genome is actually made up of coding region, roughly?
It's only 1 to 2%.
And so all this other, 98 to 99% then, I mean, a lot of people who, are just learning about the genome may be wondering, what exactly is the rest of that sequence doing?
Yes, see, originally people thought it was just junk. It was just a virus getting in and replicating itself. And in recent years, people started to look at how much of that sequence is being made into RNA, how much is being transcribed. And to everyone's surprise, more than 80% is actually made into a copy of RNA. And these RNAs have all kinds of interesting functions. For example, to regulate activity of other genes, to regulate the activity of messenger RNAs, how they are being translated, and many different functions that the RNAs have. Some of them are structural. I mean, there are RNAs that are structural components of the ribosome. And otherwise, outside of the coding sequence, there are control regions that are important to regulate the activity of each gene.
I see. So we learn in this lesson about messenger RNA, mRNA. And so what you're saying, there are actually other kinds of RNAs that can be made besides mRNA that doesn't get turned into protein?
That's right. And what was found out recently, that you remember there are 2 strands in the DNA, and the messenger RNA is only made of 1 strand that gives the information for the protein. But what is being found out now is that there are antisense RNAs, that actually the other strand of DNA can also be made into RNA. It goes in the opposite direction. It has no coding function for proteins, usually not, sometimes it does, but it has regulatory function. I mean, you just can imagine if a gene is transcribed in the other direction, then the transcription of the messenger RNA has a problem. It runs into, it's a train wreck, right? So there is a regulation of gene activity by antisense RNA. That's one mechanism.
Right. So it sounds like maybe some of the complexity of different species or the complexity of cells is in part not so much the sheer content, the number of genes you have, but maybe how you regulate all of those genes together to create something.
What we are finding out now is that the genomic regions communicate with each other. Like you can have an enhancer region that is downstream away from the gene, or even in an intron of another gene that then folds over, communicates with the promoter, and sets in motion the messenger RNA synthesis. So the whole genome is 3-dimensional. It's not just 1-dimensional series of letters. It has a lot of 3-dimensional arrangement and interaction that's very important for its function.